Hello everyone, my name is Basima. I was a master student of agricultural biotechnology and one of the Palestinian-German Science Bridge Fellows in 2017. My research was detection of metabolites from spoilage microorganisms in food products using printed electrochemical sensors, and we concentrate on studying milk spoilage by bacterial metabolism. This research took place in Institute of Complex Systems and Bioelectronics ICS-8, and it was supervised by Dr. Nawaf Abu Khalaf from College of Agricultural Science and Technology at the Palestinian part, and Dr. Alexey Yakushenko from Institute of Complex Systems and Bioelectronics at the German part. The launch-up of this research was by this advertisement on Fortune Syndrome Jülich magazine, talking about yogurt freshness and how it can be tell. So for a 3 months research period, our goal was to investigate the possibility of using printed electrochemical sensors to measure and later predict proliferation of microorganisms causing spoilage of food products produced within a package. Please think about the following question. Do packaged food expire before or after the expiry date? Do storing conditions affect the freshness of packed food and spoil it before the actual expiry date? Do we actually have to throw packed food after the expiry date? Do factories have to damage tons of packed and canned food just due to the expiry date? And also, do different types of food transporting process affect the freshness of packed or canned food? What happened to an opened canned or packed food if it were left open for a few hours at room temperature, then it was put back again to fridge? Or what will happen if you drink up directly from a packed fresh drink and then you put it back to fridge? We can come to the main questions. How would you tell if your food is fresh? Or what if your food can tell you whether it is consumable or not? This can be done using electrochemicals microchip that will be placed inside packed food, which will be connected to a server to give information from inside about the freshness conditions such as temperature, microorganisms proliferation, pH, oxygen, and etc. Here we have studied pH, oxygen, ascorbic acid, humidity, and temperature in both conventional and bioelectronical ways. The launch-up of this research was by this advertisement on Fortune Syndrome Jülich magazine, talking about yogurt freshness and how it can be tell. Each tested 1 liter sample was divided into two 500 ml samples that were incubated at 2 different storing temperatures, one of them at 4 Celsius degree and the other at 22 Celsius degree for all the held tests. For the conventional pH test, the electrode was calibrated every Monday using calibration solution. Measurements were taken for 24 days at 2 different periods during the day, where 3 test tubes were filled with 4 ml of each fresh milk samples in different conditions. The next level was for biological test, where Linux broth media with agar was prepared by dissolving 35 gram powder in 1 liter with heating, then autoclaved and allowed to cool down and poured in 5.6 cm and 9 cm petri dishes to solidify. Under sterile conditions, each test milk sample was serial diluted up to the power -8 and then 100 microliter of each dilution was spread evenly over LB agar media that were incubated at 22 Celsius degree for 48 hours, and then the colony-forming units per ml were recorded for each sample. And also the next level was for the contamination test, where 4 participants' saliva were inoculated into 4 fresh milk samples that were incubated in both conditions and tested for both pH and microbial growth. The previous tests were done conventionally and bioelectrochemically to come up with the microchip suitable fabrication. Here are some of our research results. Here you can see the pH measurement of fresh and expired samples that were stored at 4 Celsius degree and their related colony-forming unit per ml of bacteria, where the pH level for both conditions were approximately 6.75, which doesn't allow for any bacterial growth. But here you can see the difference in the pH levels with samples stored at 4 Celsius degree and that one on 22 Celsius degrees over the 24 days of testing, where the pH for both fresh and expired samples start to drop from the 5th testing day, as you can see. For the conventional pH test, the electrode was calibrated every Monday using calibration solution. Measurements were taken for, as you can see here, there were 2 types of growing bacteria according to pH level that start to grow after 3 days of opening fresh milk samples at room temperature with a pH level of 6.69. This was an airborne white to yellow large bacteria that start to decline at a pH level of 5.4, allowing the growing of another white to colorless small bacteria after 15 incubation days. Here you can see the change in milk texture during the 24 testing days at different storing conditions as milk start to become yogurt. Here the change of milk texture according to the change of pH level. And also here you can see the different change in milk texture according to the inoculated saliva, where S1 remained the same after 24 hours of inoculation. Meanwhile, S1, 3 and 4 changed very fast in the same testing period. Our conclusions are: the maximum colony-forming unit per ml in drinkable pasteurized milk sample is 20,000 per ml. The undrinkable threshold of pH change caused by bacterial metabolism is 6.65. If the pH value was continuously measured above 6.65 for at least 5 days, it alone can be used as a single measurement for milk freshness. pH and temperature sensors in a smart fridge can determine milk freshness at pH of 6.65 for less than 5 days. This with knowing that bacterial growth will accelerate during elevated temperature. Proper storing of opened milk sample. Preserve it till 30 days after the expiry date with no bacterial growth nor changing in milk texture. Inoculated microorganisms such as saliva to milk sample will enhance the spoilage mechanism even in proper storage conditions within 1 week. The number of airborne bacterial cells and their type that start to coliform in milk reflects the time it needs to spoil the milk sample. Taking into the account the bacterial colony-forming unit and the pH measurements as an indicator for milk spoilage, we had these spoilage detection categories. The first category is true positive when the colony forming unit per ml is above 20,000 and the pH level is less than 6.65. True negative when the colony forming unit is less than 20,000 and the pH level is above 6.65. The false positive when the colony forming unit per ml is less than 20,000 and the pH level is less than 6.65. And the last category is when the colony forming unit is above 20,000 and the pH level is above 6.65. In the second and the fourth categories, as an outlook to determine better the microbial metabolism in spoiled milk, both pH, temperature, and conductivity sensors measurements together should take place. In the third category, which is the false positive, this situation is very rare in a good texture milk sample. This can take place where either the milk sample is chemically contaminated or microbially contaminated, where the milk is changed to another form or textures. Our conclusion helped in the establishing of a spin-off company called Is It Fresh, and in improving the printed nanotechnology. The whole scholarship helped in transferring techniques that I used in my thesis and in transferring ideas between both sides. I also gained international experience in experimental design, sensor design, and data analysis. My master thesis was defended and approved, and the new articles are in preparation. This is my certificate in work and our group of research. Thank you for your attention.